Yeah, let's just wait for 1 minute. Hey. Okay. Hello, everyone, and welcome to the Medican Express webinar series. Today's topic is on ex vivo drug response profiling in clinical applications. We're very fortunate to have with us today Dr. Peter Martin Bruch, a distinguished young physician scientist. Dr. Bruch and his team are working at the forefront of biomedical science, bringing the fruits of lab research to directly benefit patient life quality. His research journey started in 2016 as an MD student in Professor Sasha Dietrich's lab. His early research focused on ex vivo effects on microenvironmental stimuli in chronic lymphocytic leukemias, where he won the Franziska-Kolb-Foundation Research Award for his outstanding scientific contributions in the field of leukemia research. After completing his medical studies in 2020, he continued his research work alongside his clinical duties in hematology departments at the University Hospital Düsseldorf. Currently, Dr. Brooks' work revolves around using multiplex immunofluorescence to characterize lymphoma biopsies by studying both the tumor intrinsic factors and the surrounding microenvironment. He aims to identify the predictive biomarkers that could guide therapy choices. His goal is to tailor the treatment options to the unique biological profiles of each individual patient by integrating functional profiling, multi-omic analysis, and advanced mathematical modeling. Dr. Brooks and his teams are paving the way for biology-based individualized treatments to enter clinical practice. We're really excited to hear from Dr. Brooks today as he shares his insights on how ex vivo drugs response profiling is making an impact on the clinical decision making. So please join me in welcoming Dr. Peter Martin Brooks.
Yes, thank you very much for the kind introduction. I'm very delighted to be here and to present to you mostly the work we've done in the past, but also putting it a bit into context of the work other people have done. Because obviously this builds on a strong foundation of a multinational effort to bring drug screening or functional precision medicine into clinically relevant application. So as you probably all know, drug response is a complex interplay that is defined by a lot of cell intrinsic factors as well as microenvironmental components. And we try to read out some of these cell intrinsic factors using genetic profiling, transcriptional profiling, and then group our patients to responders to define the most effective therapies. And we try to increase and use more and more biomarkers. But in the end, these are all indirect measurements of what we actually want to predict the response to anti-cancer drugs. And basically, what we tried with functional precision medicine is to directly profile the end outcome that we want to see. Multivariate modeling in the past has revealed that different factors influence drug response, and this is vastly different for different drugs, interestingly. There are some drugs, such as classical chemotherapies, here, for example, shown fludarabine, which are mostly influenced by genetic factors. And it's here, for example, TP53 as a resistance mutation. And for these drugs, you can nicely use genetic factors. However, other drugs, such as ibrutinib or idelalisib here, for example, which are kinase inhibitors, are mostly regulated by other factors, and genetic roles didn't really play a major role in them. So if you try only to categorize your patients into responders and non-responders by genetically profiling them, you're bound to fail, at least from the genetic annotations we know thus far. And their genetic, their gene expression, methylation, and demographics largely impacted the response of these drugs. And then what other people have done before, and here's an exceptional paper that I'm sure many of you are aware of, is really guide the therapy of relapsed patients who don't have a standard of care treatment anymore by selecting the most effective drug based on drug response profiling. And here it was a great finding that they could identify exceptional responders, which selected the drugs based on the profiling ex vivo, and they had vastly long response to these drugs, so that we hit really the right target there for this specific patient. And then that led to a great outcome, which was here in mostly TNHL, so a very adverse risk cohort, and the patients that the treatment that they had before, which was not informed by functional precision medicine, was ineffective. And this led us to the question whether we can do the same thing inside clinical routine in standard of care settings. So not in situations where we don't have any idea what we want to treat the patient with, but in settings where we definitely know standard of care treatments and there are fixed regimes, and whether we could there identify responders and non-responders. And here I have to especially thank Nora Liebers, who was a person behind the clinical trial, the SMARTrial, which was performed in Heidelberg. And she led the whole clinical part of this, assembled the patient cohort and helped with the data analysis greatly. And then what we did, we had a very open trial setting. Any hematological malignancy was open. There had to be a treatment indication. So we wanted to do a treatment, measurable disease burden to judge the therapy response. And the key factor was that we somehow had to get our hands on viable tumor cells. This could be from blood in leukemia. This could be from biopsies. For example, in lymphoma, this could be from bone marrow aspiration. Any kind of cell, any kind of tumor cell was suitable. We had exclusion criteria. For example, if the patient couldn't receive therapy or they had already received therapy just before the biopsy. And then these samples we took from the patients were screened with a broad profile of drugs. The target being to actually screen every sample that was also applied in the clinical routine, and additionally have some investigational samples for those patients that don't respond to their therapy, have a backup option to suggest alternatives. We had ready-to-use plates, which greatly alleviated the resource limitations, because we all know that a patient that you want to include into your clinical trial never comes in Monday morning at 8 a.m., but they always come in Friday afternoon. And it's really annoying if you then at Friday afternoon have to start setting everything up. So we had ready-to-use plates that could be, after isolation of the mononuclear cells directly, the cells could be pipetted on them. They went into the incubator, and then you had 48 hours to take care who's coming in on Sunday night to read out the plates. We had, as in any clinical trial, a follow-up schedule, which was very intense and short-circuited in the beginning of every week. And then 3 months, 6 months, and 12 months follow-ups. This led to the recruitment of 80 patients, which was the target size of the cohort. 80 patients were enrolled, and unsurprisingly, the two most common leukemias were the main disease groups. So AML and CLL were the largest disease groups, but we did also include a number of lymphomas as listed here. For the primary endpoint analysis, all patients were eligible. Our primary endpoint was the feasibility, so getting results within 7 days. And over 90% of samples could be read out within the time frame, and drug response could be reported back to the treating physicians directly. This was possible due to an automated data analysis pipeline where right after the readout in the lab, the data went online, went through, was compared to all other patients, and was reported back. And the secondary endpoint was the association of in vivo and ex vivo drug response. 64 patients could be considered for this endpoint, as some of them were excluded mostly due to the lack of measurable response in vivo. And if that couldn't be evaluated, it's very difficult to associate it to anything. We concentrated then here, or I'm concentrated here in the talk on the chemotherapy cohort because it was the most homogeneous cohort to investigate. And what we interestingly found that if we performed an elastic net regression to see what drugs mostly, so what drug responses ex vivo mostly defined drug response in vivo, was that the vinca alkaloids here, vincristine, were highly predictive for their response and other chemotherapies as well. And this was associated significantly with event-free survival, which was basically defined as a progression or death. And you can here see that these samples were selected in a very high proportion of cases in the model, so they strongly impacted response. Now, we also looked at this in a more closely fashion here, for example, with Vindazine, and you can see that the response of the ex vivo drug response of the patients that in the clinic responded, which are here termed as R, so responders, was a lot better. So the viability of the cells was lower, whereas the patients that had a progressive disease in the clinic were almost, the cells weren't dying at all in the lab as well. Now, this is a very small number of samples, especially in the progressive disease side, and that's good because we treat a lot of our patients very well already. However, for the statistics and to actually gain any knowledge, it's a bit difficult, which is why we had a validation cohort, which consisted purely of acute myeloid leukemia. So it was selected to be as homogeneous as possible. All samples were treatment naive, and all samples received daunorubicin and cytarabine, the standard of care induction therapy, after their sample was taken. We performed drug response profiling here with a more targeted panel, as we knew in advance what the drugs the patients received, and we investigated 23 single drugs, as well as daunorubicin, cytarabine, and venetoclax plus Vidaza, the two standard of care treatments, in a combination matrix. And for those of you that are not familiar with AML, AML is strongly driven by genetic factors, and the drug response is known to be influenced by those genetic factors. So there's risk classification, grouping the patients into favorable, intermediate, and adverse risk patients, considering their survival. Which is why the group was split up to be representative, and we had in all 3 risk groups both responders and non-responders, as you can see here. And then in this validation cohort, we again associated the response of the ex vivo drug response profiling with the in vivo therapy response. And we again saw that in this chemotherapy-treated cohort, chemotherapeutic drugs were strongly predictive, and interestingly, not the single drug treatments of daunorubicin and cytarabine weren't predictive. However, the combination matrix of both drugs that the patients also received together were predictive for their therapy response. And And then the main question is whether this holds true beyond genetic factors, because one could argue now this is only a TP53 mutation we read out in the end, which is why we next investigated this based on the 3 genetic subgroups that we know are influencing the therapy response in AML. And what we observed is that especially in the adverse risk patients, so those patients that have a high risk of not responding, that definitely need an allogeneic stem cell transplantation, we could differentiate between those samples that would respond and wouldn't respond in the clinical setting to their treatment. And this then also translated into a significant survival difference for these patients based on their ex vivo drug response profiling. Now, the interesting part is that our results suggest that there is a knowledge to be gained by functional drug response profiling beyond what is already known on genetic markers, at least in AML. The difficult question is, where do you draw the line and how do you really optimize the response? Thus far, in clinical setting, it is only done for refractory patients, but I know that currently new clinical trials are being set up to answer these questions, to really profile the patients in an interventional setting and find out whether a functional precision medicine guided therapy can improve their therapy efficacy beyond what we already know in genetics. Now, this obviously needs to be integrated also with other data, because I've talked about cell intrinsic resistance mechanisms here. However, what most, at least most assays miss currently is the microenvironmental compartment, because we can profile parts of it in microscopy or FACS-based drug screenings. However, it's difficult to account for the heterogeneity of the tumor microenvironment in drug response profilings ex vivo. And unfortunately, that's all I can show you already, because the new results that would perform interventional studies in this case aren't ready yet to be shown, but I hope that in the next future we can see those trials going online, and hopefully in the a bit more distant future, patients will actually be treated differently based on their drug response profiles, to actually treat patients really on their single drug response profiles and not on indirect measurements such as genetic changes. I have to acknowledge all the contributions here and thank especially the patients who contributed here, because it wouldn't have been possible to do any of this without patients that are ready and prepared to take part in scientific research, but also all the researchers here, mostly Nora Liebers and Sascha Dietrich from the lab in Heidelberg and now in Düsseldorf, Wolfgang Huber and Junyan Liu, who at that time were at EMBL, and Junyan Liu has now moved to the University Hospital in Heidelberg, as well as Thorsten Sens in Zurich and Axel Benner at the DKFZ. Thank you all very much for your attention, and I'm happy to take any questions. So, I'm sorry, I think we can't hear you just yet.
Okay. Yeah, let me see if any questions raised in the okay.
We don't have the chat, so I can't say whether there are any questions there.
I actually myself have a questions. I saw your paper in that Nature paper. You using the ATP based method to screen the drugs against the patient's samples. Do you think the choice of the assays you used will affect the predictive powers of your response? Like, because some of the drugs that maybe have a long-term effect of targeting the cancer cells. Others maybe have very quick onset, like maybe cause DNA damage and the cell will just die, and others maybe trap the cells into like mitotic commitment, not into the mitosis, and just stay there for some time? What's your...
Absolutely. I think that has a vast impact, and I think that's one of the big challenges in actually rolling out functional precision medicine. There are two aspects to your questions in the end. The one would be the assay, and I think, yes, it makes a difference. This trial was based on ATP readout. The rationale was to have a very robust and easy to use assay that can be done in the clinical setting. We have done other trials where we have used, for example, microscopy-based screenings, and for example, the people in Zurich and other labs have used microscopy-based screenings, for example, in Vienna. I think the big advantage of microscopy or also FACS-based assays is that you can read out both tumor subpopulations and microenvironmental effects more thoroughly. I'm not entirely sure which will in the end be the best trade-off of usability and robustness, and on the other hand, exactness of readout. I think ATP has its limitations. So I think if we really want to go for in-depth readout that captures subpopulations, especially in diseases such as AML, where that's important, I think we need to go for some kind of single cell assay, and that can be FACS or microscopy.
Right.
I don't know yet. The other thing is the duration of observation, and I think that is also extremely crucial. You said many drugs work quickly, and many drugs have a long-term effect. I think that's something we have really a hard time comparing. In the end, you would have to do both. You would have to do a continuous measurement and then compare that. What we found to be helpful is not comparing the drugs directly to each other, because then you're extremely biased by the different concentration ranges and kinetics of response. We found it helpful to first compare the patients to each other, so the patient responses within one drug, and then that measurement you use to compare the different drugs. Which is why in the end, or how in the end we selected drugs in cases where we actually had therapy-resistant patients and we needed a functional precision medicine-guided therapy. This only works if you have a large reference cohort, and I think that's also difficult because that reference cohort is really biased by how you select your patients and how you did your drug screen. So all of these things, I think, hinder the implementation in clinical routine.
Okay, thank you. Yeah, we have a more question coming from the audience. The first one is from Lisa. The question is, how do you decide what concentration to use for those drugs? Do you follow any
Yes.
like protocols or look into a database?
Yeah, in the end, that's I can only answer what we did. And I know that other people did it differently. I know people have used just a standard concentration range and kept it the same for all drugs. And have had very robust readouts with that. And for example, the paper, I think, from the Vienna people were just that. And that's perfectly fine. We decided differently and decided on previous drug screens we have performed. And we had the luck that we, just before this clinical trial, finished extremely large drug screen with, I think, 500 or 600 drugs in a limited number of patients. So we had the chance to select from there, where we had an extremely broad range of drugs, the most effective one, and then take that into the clinical trial. I don't think there's a perfect solution. I think if you're limited in the number of concentrations you can investigate, as we were, because we had limited cell number, I would rather go for a drug range defined on previous knowledge.
Okay. Thank you. Yeah, the next question from Durden. Could you tell a bit more about the data analysis pipeline as in how you classify cells in dead or viable cells. And do you use like features from imaging data and then training in a machine learning based models?
That's a very interesting question. It's difficult, or it's different for different projects. A clinical trial we performed was, as has been already mentioned, ATP based. So you had per well a readout of how much viability or how much metabolic activity rather was present, and then we normalized that to untreated controls and compared that between patients. In more sophisticated investigations, such as bispecific antibody drug screening, which has been published in Blood Advances, we performed imaging, and there we used a machine learning approach where you previously defined on both marker expression and cell morphology the viability, and you defined viable and dead cells, and then that machine learning model learned features and took that over the whole cohort. That's a lot more work intensive, and I think it's not as scalable. And one has to see how modern machine learning approaches perform than if you try to upscale this to more patients, more samples, especially different diseases. That's going to be difficult. So I don't have a perfect solution for the future.
Okay, thank you. Yeah, then the next question is from Lisa again. This one's quite technical. She's asking, is the plates pre-plated with the tested drugs in there? So basically asking you how you handle those screening plates. Can they like being kept for quite a long time and transported around?
That's a good question. Again, I can only say what we did. So we had a test run of pilot phase, and then for the clinical trial, we freshly prepared everything in one batch and froze everything down at -20. And then we finished off the clinical trial within a year. We did repeated samples at the beginning, at the middle, and the end, and compared these to each other. And there we saw that the drugs were very stable, and everything worked very well. I can't say whether it holds longer than that year we did. I mean, the trial has been over now for a while, so we could test again with the old plates. I would be in favor of keeping them not longer than a year or two, if possible. Initially, we tried freezing them at -80. We had some problems with the consumables and the plates, which is why we in the end went for -20, and that worked for the year very well.
Okay, thank you. Yeah, and we have 3 questions from Hugo. And the first one is, how does your functional drug screening IC15 data is correlated with the MRD status and MRD amount upon inductions and consolidation therapies in AML?
So I guess he means MRD status after induction. Unfortunately, this is one of the limitations of our cohort. It was a registry-based cohort, and no centralized MRD measurement was taken for these samples. So I can't answer that question. The information we had were response by flow cytometry, and that's the annotation we have here we had here that I've shown in the Kaplan-Meier curves of responders, non-responders. That were the classical clinical cutoffs. MRD information, unfortunately, wasn't available.
Okay. Yeah, the second question from Hugo is that, how do you envision the next generation of a functional drug screening will emerge? And what's your opinions on the endpoint analysis and type of platform of measurements, or on the 3D modeling of the microenvironment? Yeah, I'm a bit curious as well. So what's your opinion? For example, is that suitable for like an organoid culture for this kind of for solid tumors? Yeah.
So I think that's very depending on the entity you talk about. Personally, I think we are going to move towards microscopy-based assays, because I think they allow you to have both spatial readout. They allow you to have also different effector cells in there, as we've shown in the Blood Advances paper. And I think they allow you to have a more finely grained single cell, at least partly, readout. As well as a readout of different factors. So you can't only do dead alive, but you can also have multiple other markers. So I think it's going to be microscopy based. I think it's going to include somehow the microenvironment. And I think for solid cancer, it's going to include some kind of 3D model, although I'm not an organoid expert and I'm not sure what is going to be in the end the best way. I think, although I've now said that everything is going to get more and more complicated, I think that's also our biggest limitation, because as we've seen, we can already, with the simplest approach of all, with ATP, beat pure genetic drugs, pure genetic profiling. And now everyone is talking about how can we make it more complicated, more complicated, more complicated to have the best predictive power. But that will never be feasible in standard of care settings outside of the university hospital, because even the ATP assay takes so much resources to do it that no peripheral hospital can do it. So I think while we are getting better and better at what we do, we're at the same time killing ourselves because we're making it so difficult to roll out. And I'm not sure. I think in the end, the only way that this can work is that if some academic or industrial consortium or company sets up one solution that they sell, because streamlining this and making it comparable between 100 different hospitals is going to just be impossible.
Thank you. Yeah, and Hugo's third question, I think, is great for the physician scientists like you. So how far are we to make this kind of analysis or this kind of a diagnostic approach into like standard care in clinics, like for like de novo diagnosis? And in your opinion, what would be the major bottlenecks for this for the next couple of years for the clinical implementations?
Yeah, in the end, I think the bottleneck is that we would need one very large interventional randomized trial that compares standard of care treatment to functional precision medicine guided therapy. And no pharma company really has interest in that, because you would have to include different drugs, and most of these drugs are also already approved and from different companies. So there's not one funding body or agency that sets up this clinical trial, because clinical trials cost like tens of millions of euros. And especially in this complicated setting where you would have to ship the fresh sample, have it centrally analyzed, get the results back, and then treat differently. I think that's going to be extremely expensive, and I'm not sure whether any academic partner can get the money to actually do that. I think it would be great, but I think it's difficult. And the pharma companies themselves, I think, are limited because they don't have access to patients sufficiently to actually do this. Because they are still reliant on having a worldwide network of partly peripheral hospitals, partly university hospitals, and not every one of those has the facilities to actually do drug screening or even perform single cell suspensions that they can then ship overnight. So I think we have some limitations which are not on the scientific side, unfortunately. And I wouldn't know how to solve these, but they are what's holding us back, in my opinion.
Okay. Thank you. Yeah. The next question is from Carjo. How to decide dose of existing drugs for other indications, ex vivo studies, like tissue organ bath studies? Yeah.
So So if I get this correctly, the question is how do we move from our concentration in our assay to other assays in maybe other entities?
Yep.
I think it's difficult, as is moving from in vitro to in vivo. I know that pharma companies have like people only working on that and how to calculate different concentration that should be equivalent in different settings. I don't have a good answer to that, to be honest. The only way we have found it to be working for us is to do small pilot experiments, run a small batch and get experience within the assay that you actually use. I know there are computational models that the pharma companies use, but I don't have any experience with that.
Okay. Thank you. Yeah. The next question from Dudens again. Have you tested any correlations of your ex vivo based assays with the clinical drug response data?
Yes, that's what the validation cohort was meant to show. The problem here being that we don't have, or we rarely have in vitro or in situ, so patient drug response, therapy response as a continuous variable. We usually have our cutoffs of what we believe is a good response or a bad response. Obviously, we have, for example, an AML percentage of blasts after therapy, but that is strongly biased by percentage of blasts before. So we don't have a perfect continuous variable after therapy that we could directly correlate as a scatter plot with our data. What we did, therefore, was the binarized responder non-responder, and that's what I've shown that we do see a strong difference there.
Thank you. Yeah, the next question is from Alexander. I think he's mentioning a papers. Okay, are you planning to integrate the response of different subclones in the drug screen for clinical setting? As in the I think he referenced a paper, the Nature Cell Biology paper.
I am aware of that paper. That's a paper from our lab. So thank you for not referencing any other paper that I'm not aware of, because then it would be difficult. Yeah, it's difficult. As I said, I think microscopy-based or FACS-based assays have a strength in targeting tumor subpopulations. And there you can actually observe also distinct subpopulation-specific drug responses. I think actually using this in the clinic is very challenging because you can't like treat 3 different subclones in the same patient with different drugs at the same time. What you can do is obviously you can see whether there is one drug that works for all subclones or subpopulations rather, and then try to use that. We haven't done that in the clinical setting. As I've mentioned, that's very limiting in cells you have available and in resources. So that is one of the questions that future trials will have to answer, and I'm really keen to see what comes out of that.
Okay. Yeah, we have more questions that actually came in. Okay, the next one's, do you have any advice for working with DMSO solubilized drugs with the poor aqueous solubility? It's quite technical, this one.
Yeah, that's perfectly fine. I'm for these technical questions, I'm better than for the future because I don't know the future. So we diluted all drugs in DMSO. Because we found this to be a huge problem. You can't solve all drugs in water, in our opinion. So we just solved everything in DMSO, and that worked very nicely. So comparing the DMSO only treated sampled cells to non-treated cells, the viability drop was extremely low. You obviously have to take care of the DMSO concentration. I think we limited it to 0.3% in this trial. We have done higher, I think 0.5 in other projects, and observed no problems there in the 384 well format. In the 1536 well format, we have observed problems with that concentration. likely because the cells are more vulnerable in these finely grained differences, because then the temperatures drop more quickly and stuff like that. So there we use even lower DMSO concentration.
Okay, thank you. Yeah, we have another questions from the coder again. So I think she or he's asking, yeah, he's a PhD scholars in respiratory pharmacology. So she or she is asking which ex vivo studying would be like best for testing the efficacy of novel compounds for the respiratory diseases, in your opinion?
Well, I think respiratory diseases are a very broad field. I think that's a very difficult question to answer in general. I think the main question is, what do you want to answer? All drug response profiling studies I have been involved in were at least partly had the goal of killing the cells, because I work in oncology. We just very stupidly want to kill. And I guess that's not in all respiratory diseases the case. Usually you want to have better expansion of the lung or something. So I think that you have to very carefully think about what that drug should do and then how you can mimic that. So I think it's very difficult for me to answer. Pulmonary fibrosis, I now read. I would have to think about this. Obviously, it's more of a long-term effect, the fibrosis. And I'm not sure whether drug screening in the high throughput setting that we've performed here is the right way to go. It might be that you rather need a long-term assay culture that actually observed the fibrosis of those cells. But anyways, I think if it's fibrosis, I think you're not going to go with ATP and you're not going to go with FUX, but you need some microscopy-based readout that also sees this difference. But maybe for the last question of Kayal, there are many universities that have drug screening core facilities, and they're usually very open, in my experience, to approach them with different ideas and different thoughts, and they usually have a very broad experience with different projects. So you would best check locally or internationally and see whether one of these core facilities has a model system that is set up for you.
Okay. Yeah, thank you very much, Dr. Bruch, for such an interesting talk today.
We have two more questions that just came in.
Oh yeah, okay. Yeah, here goes again. Do you know what is the effective drug concentration that leukemic blasts experiments in the patient's bone marrows during the inductions chemotherapies? How far are we for the use the IC15 values into the TCPS functional drug screening ex vivo?
I can't answer that off the top of my head, to be honest. I know that there's data investigating the drug concentrations in the bone marrow. As far as I'm aware, it's very heterogeneous. So it's very difficult to say how well this corresponds to the ex vivo settings. Unfortunately, I can't answer this off the top of my head. I'm sorry.
Okay. Yeah, the last question is from Dudens again. Could you give some insights for the evaluations of the effect of chemotherapies versus immunotherapies?
Well, only that it's extremely difficult. Chemotherapy is the nice and easy part, to be honest, because it works quickly and it works without effector cells present. So that's quite easily profiled, to be honest. Immunotherapy can be investigated in ex vivo profiling. For example, we did this with bispecific antibodies, but you need vastly more complex model systems. So you need effector cells that are present, and then that if those effector cells are sometimes autologous because you can get them, then it's great. But if they're allogeneic or from a cell line, then you have HLA effects, and you have to somehow account for those. And then your drug also sometimes works on the effector cells, and it's an extremely complex interplay. However, since we obviously move into more and more towards immunotherapies in the clinic, it's extremely relevant. And we have shown that it can work if you in a very targeted fashion, think about your scientific question and then optimize your assay culture for that. So, for example, we've set up a assay culture that has been recently published in Blood for CAR T cells and bispecific antibodies at the same time, and their effects on lymphoma and leukemia cells. And that's possible, but it's even further away, or a lot further away from clinical application, in my opinion.
Okay. Yeah, I hope that answers all your questions. Well, yeah, first of all, yeah, both of them are thanking you for your answers, and yeah, wish you the best with your research forward. Okay. Yeah, thank you very much, Dr. Bruch, for such an interesting talk today. And thank you all the audience for coming, attending this webinars. There will be more exciting MC webinars coming soon this month. Our next speaker is from the Australian's National University. He's going to talk about how our innate immune receptors influence the tumor development as well as fighting the infectious disease. So please stay in tune for that. At Mac Express, we're committed to fostering scientific communications and accelerating the pace of scientific discovery. Thank you again for joining us today, and stay connected. And I look forward to seeing you all very soon. So goodbye for now. Thank you.
Thank you. Goodbye.